Okay, imagine this. Buildings that don't just sit there, but actually clean the air around us. Sounds pretty wild, right?
Eh,
Like something out of science fiction.
it really does.
So today, we're doing a deep dive into some really fascinating new material on living materials, capturing carbon with photosynthetic bacteria. Basically, our mission is to figure out how scientists are actually designing materials that breathe in CO2.
Right, materials that pull carbon dioxide right out of the air and, turn it into something stable, something useful.
Which brings us to the big question for today's dive.
Can we really make our buildings, our infrastructure, into giant carbon sinks?
Exactly. This research we looked at, it seems to be getting us closer, mixing biology and engineering in some pretty surprising ways.
It's definitely pushing boundaries.
So with that question hanging there, let's maybe start with the basics. What exactly are these living materials?
Okay, so the sources define them as essentially conventional materials. Think polymers, gels, but they're combined, integrated with microorganisms. We're talking bacteria, algae, sometimes fungi.
Microscopic life inside the material itself.
Precisely. And the whole point, the goal, is to use the natural processes of these microbes, their metabolism, to give the material new, useful properties.
Okay, that makes sense. And the specific example we're focusing on comes from ETH Zurich, Professor Mark Tibbet's lab.
That's the one, yeah. They've created this really interesting photosynthetic living material.
And the key ingredient, the living part, is cyanobacteria, blue-green algae.
Cyanobacteria, the research calls them, ancient and incredibly efficient photosynthesizers.
Ancient, right. Billions of years old.
Something like that. And they're great because they can use even pretty weak light to make biomass from CO2 and water. Very effective little carbon converters.
So how do they get these bacteria into a usable material? You can't just, I don't know, smear algae on a wall.
No, not quite. The clever part is the packaging, as you might call it. They embed the cyanobacteria into a special hydrogel.
A hydrogel. A gel mostly made of water.
Exactly. A water-rich gel made from cross-linked polymers. And it's printable, so you can 3D print structures with these living cells inside.
Okay. Printable and alive.
Yeah. This hydrogel matrix keeps the bacteria happy, lets them grow, and most importantly, lets them actively capture carbon while being part of a structure.
But here's where it got really interesting for me in the source material. It's not just about the bacteria growing and making biomass. It captures way more CO2 than that, right?
Yes, that's the crucial part, this dual carbon sequestration they talk about. It goes beyond just biological growth.
Dual. So 2 ways it captures carbon.
Correct. While the cyanobacteria photosynthesize, they actually change the chemistry of the hydrogel immediately around them.
Okay. How so?
Well, the process leads to the formation of solid carbonates. Think minerals like lime, calcium carbonate. These minerals precipitate right there within the gel.
So they're basically making tiny bits of rock inside the material using the captured CO2.
That's a great way to put it, yeah. And those minerals, the carbonates, they lock up the carbon in a much more stable, long-lasting form than the organic biomass alone.
That sounds permanent, or at least very long-term storage.
It is. Much more stable over geological timescales, potentially.
And this mineral formation, it must affect the material itself.
It does, and that's another clever aspect. These minerals that form, they actually reinforce the material mechanically.
So it gets harder over time.
Exactly. You start with this relatively soft 3D printed hydrogel structure, and as the bacteria work and the minerals form, the whole thing slowly stiffens and hardens.
Wow. It builds its own reinforcement using atmospheric carbon.
Pretty much. The ETH study actually showed images where you see these green, soft structures becoming rigid over about 30 days. It's a self-reinforcing process.
That's really elegant. What does this living material actually need to live and do its job? Is it high maintenance?
Surprisingly, no. According to the research, it just needs sunlight. Doesn't even have to be super strong carbon dioxide from the air, obviously.
Right.
And basically, artificial seawater with some common nutrients, things like nitrogen, phosphorus. It's quite minimal.
Almost like a very functional houseplant.
Kind of, yeah. But one that's potentially part of a building. The hydrogel itself is key here, too. It's designed very carefully.
How so? What makes the hydrogel special beyond just holding the bacteria?
Well, it has to be permeable, right? Light needs to get in for photosynthesis, CO2 needs to diffuse in, water needs to be available, nutrients need to reach the cells.
Okay.
And it needs to keep the bacteria distributed evenly so they can all function well. So the polymer chemistry and structure are really important.
And I guess the physical shape you printed in matters too. Using 3D printing suggests they can control the form.
Absolutely critical. They used 3D printing to optimize the geometry. Think about creating structures with a really large surface area.
To maximize contact with air and light.
Exactly. Better light exposure, better nutrient flow. It's all about creating the best possible microenvironment for those cyanobacteria.
And did it work? How long could they keep this system going?
The results were impressive. They managed to keep the cells productive, continuously capturing carbon for over a year in the lab setup.
Over a year. Wow. Okay, so let's talk performance. How much CO2 are we actually talking about?
The lab tests showed the material continuously binds CO2 for over 400 days.
Continuously for 400 days.
Yeah, and most of that carbon, crucially, ends up in that mineral form, the carbonates.
Right, the stable form. Do they have numbers?
They do. They quantified it at around 26 milligrams of CO2 captured per gram of the material.
26 milligrams per gram. How good is that? Is that a lot?
Well, putting it into context is important. Compared to many other biological carbon capture ideas, that's significantly higher.
Okay.
And interestingly, it's actually comparable to some chemical methods, like the mineralization you get with recycled concrete. That's often around, say, 7 milligrams of CO2 per gram. So 26 is quite substantial.
So for a living system, that's punching above its weight, potentially.
You could say that, yeah. It's showing really promising efficiency in these lab conditions.
So what's the vision here? If this works so well in the lab, what are the hopes for, the real world?
The researchers see this as a potentially low-energy, environmentally friendly way to bind CO2 right out of the atmosphere. Not capturing it somewhere else and storing it, but binding it where it is.
Okay, low energy because it's powered by sunlight, mostly.
Primarily, yes. The energy input is mainly light.
And the applications. Where could we use this?
Well, the big idea, the exciting potential, is using it in construction. Either as a building material itself, imagine bricks or panels that store carbon.
Living bricks.
Or perhaps more immediately feasible as a coating, like a living paint or facade layer on buildings.
So the entire surface of a building could be actively capturing CO2 throughout its whole life.
That's the vision. Turning our cities, or parts of them, into active carbon sinks, buildings that literally breathe in CO2.
It's a pretty radical shift in how we think about buildings, from inert structures to active environmental participants.
Exactly. It's still early days for large-scale use, obviously. There are challenges to scaling up.
Sure. Lab results are one thing, real-world construction is another.
Definitely. But what's fascinating is that architects and designers aren't waiting. They're already playing with these ideas.
Really? Like actual projects?
Yeah, the research seems to have sparked immediate interest. There are a couple of really cool examples mentioned in the sources.
Okay, tell me about them.
Well, first, there was an installation at the Venice Biennale called Picoplanktonics.
Picoplanktonics, catchy.
It was by an ETH doctoral student, Andrea Schindling. And what she did was pretty amazing. She scaled up the production from, small lab samples to actual room-sized objects.
Wow, okay. What'd they look like?
They were described as two objects resembling tree trunks, one almost 3 meters tall, made entirely from these living building blocks.
So essentially giant living sculptures made of this hydrogel.
That's right. And they even estimated the carbon capture potential.
Oh, yeah.
Each trunk was estimated to bind up to 18 kilograms of CO2 per year.
18 kilograms. How does that compare to something natural?
The comparison they made was pretty striking. It's about the same amount of CO2 that a 20-year-old pine tree would absorb in a temperate climate.
Per installation, that's significant.
It is. And apparently they're monitoring and maintaining it right through November this year, so gathering real-world data.
Fascinating. And you said there was another example.
Yes, at the Triennale di Milano, a project called Daphne's Skin.
Daphne's Skin, okay.
This was a collaboration, Maid Studio and Dalia Drancyk, and their focus was slightly different, looking at these materials for future building envelopes, skins for buildings.
How did they approach that?
They worked with wooden shingles, but they encouraged microorganisms to form this deep green patina over them.
So using the microbes to create a living surface on wood.
Exactly. And the idea was to reframe something we might normally see as decay, like moss or algae growth, as an active design element.
So embracing the microbial life aesthetically.
Right. A living patina that not only changes the look of the building over time, but also actively binds CO2. It was part of a larger exhibition called We the Bacteria, Notes Toward Biotic Architecture, which also sounds fascinating.
Read the bacteria. I like that. It really challenges the idea of buildings as sterile objects.
Completely. It suggests a future where architecture collaborates with biology much more deeply.
What an amazing area of research. So just to recap this deep dive, we've seen how scientists are creating these living materials, embedding cyanobacteria in hydrogels.
Right, materials that capture CO2 not just by growing, but by forming stable minerals within the structure itself.
Which makes them potentially much more effective for long-term carbon storage.
And with real potential down the line for applications in our buildings, our cities, turning infrastructure into carbon sinks.
It really is a mind-bending concept, which leads us to that final thought for you, our listener.
Yeah, if our future cities, our buildings, could genuinely breathe, could actively pull carbon from the air around us, how does that change things?
How does that fundamentally change our relationship with the structures we build, with the air itself? What kind of possibilities open up if we start designing and living amongst living buildings?
It's a huge question about design, sustainability, and maybe even our connection to the planet. Something to definitely think about.